to note about the anticodon is that it is complementary to the codon on the mRNA. Now, if at this position there's another codon, a different one, let's say it is proline, the tRNA that has to come to the ribosome must have an anticodon that is complementary to the codon for proline. And on this end of the tRNA, it must carry proline. So let's focus on this one. We want to understand what amino acid activation is or what tRNA charging is. tRNA charging is a process of preparing the tRNA to ensure that it binds to its corresponding amino acid. And by that, I mean the amino acid that's going to bind to this tRNA is, in this case, is methionine. Why? Because the codon that this tRNA is carrying is complementary to the codon for methionine. If this codon was for proline, tRNA charging means this tRNA must covalently bind to an amino acid proline. Why? Because that amino acid that is bound to tRNA, the corresponding anticodon on the mRNA, that is a codon, is specifying the message proline. Now, let's say, for example, after this codon, the next codon is CAU. Now, I've kept a distance here, not to say this is how the distance is, it is just for demonstration. I want us to understand what charging the tRNA is. So let's assume the codon that comes after AUG, and like I said, you must ignore this space because these codons are sequential. It's like writing a word. If you're going to write your name and then you're going to put a space between the letters, the message won't be, won't be your name, to be something else. So I've put a space just to demonstrate. So let's assume we have the next codon after AUG is CAU. Now, the amino acid CAU is histidine. This is the message that should come. This is amino acid that must be brought to the ribosome. This histidine does not just come to the ribosome on its own. I keep on mentioning the ribosome because we understand this process of translation is taking place in the ribosome. And that is what I'm trying to say. If at this point we have CAU, this codon is reading or should give us the message histidine. Histidine is an amino acid. How is this amino acid going to be picked from the cytoplasm? Remember, in the cytoplasm, there are all those 20 amino acids. But this time around, this codon is calling only for histidine. So meaning histidine has to be selected from the pool of other amino acids in the cytoplasm. So what's going to happen here is that a tRNA is going to be charged. The charging of a tRNA, we'll look at that in the next slide or so, is also a process. But here, the take-home message is that histidine is going to be selected from the pool of other amino acids. It's going to be attached to its respective, to its corresponding tRNA. And by corresponding tRNA, what I mean is if histidine is bound on the 3' end of the tRNA, and we know this is the acceptor arm. It has got a marker, the ACC sequence. This is where the amino acids bind. It means that this tRNA is carrying the anticodon that is complementary to the codon that is sitting on the mRNA. There's no way this tRNA is going to complementary bind to the codon for methionine. No. This complementary base pairing is made possible and allows for this tRNA to bind stably. If you switch these two tRNAs, in this case, you bring histidine here and you bring methionine here, you can already see that there will be no complementary base pairing. So one of the reasons why only the tRNA carrying the amino acid for this codon will complementary bind is because of this anticodon-codon relationship. It is specific. Of course, we're going to see in the next few slides that the third position sometimes can allow for a mismatch. But at this point, our interest is to understand amino acid charging or tRNA activation. What is it? It is just a process by which the tRNA complementary binds to an amino acid. At what position on its 3' end, which is referred to as the acceptor arm. This tRNA is going to bring in this amino acid into the ribosome. And how does it do it? It is going to use its anticodon to complementary bind to the codon on the mRNA. Keep in mind, this anticodon-codon relationship must match. That is what complementary base pairing means. And you can see, if on this codon we have a C, C base pairs with guanine, G. A base pairs with uracil. We know in this is mRNA, we don't have thymine. That is why where there is supposed to be a T, we see a U because we are dealing with RNA. And where there is a U here, it complementary base pairs with an A. This is complementary base pairing. This is what allows for this message, which is supposed to be histidine, to be supported by an amino acid, histidine. So tRNA functions as an adapter. So now the charging of tRNA. How does this tRNA pick histidine? Of course, it picks histidine because it is being guided by what is sitting next on the mRNA. That is to say, if this tRNA has interpreted the message on this codon, and the next codon is CAU, in the machinery, what should come in is a tRNA carrying histidine. Now, how does this tRNA associate with histidine? And then now we're going to look at the process of charging the amino acid. All this is done before initiation should start. Okay, so now what is charging of the tRNA? So the first step is the amino acid itself has to be selected from the pool of other amino acids. Remember, this process is taking place in the cytoplasm. And for that reason, we need to select the respective, amino acids. And in our case, we are dealing with a codon that is calling for histidine. So let's assume this is histidine. Histidine is going to react with the ATP to form a high energy molecule or high energy bond. Histidine, so this is an amino acid and it can be any amino acid. Even the first one was methionine. So meaning that if the tRNA carrying methionine should come to the amino acid, to the ribosome, the first thing that should happen is that the amino acid methionine must react first with the ATP. This is the first step in tRNA charging or amino acid activation. The amino acid has to be selected. The selection process itself involves the reaction between the amino acid and ATP. This reaction is catalyzed by amino acid tRNA synthetase. Okay, these are the enzymes that catalyze this reaction. And what you're going to have is an amino acid AMP. We're going to have the pyrophosphate being lost. So now, in our case, we have a codon for histidine. What that means is that the amino acid histidine is going to react with ATP. Now, remember, each amino acid should have its own amino acid tRNA synthesized. Why? Because it has to be uniquely selected. If collectively we just have amino acid tRNA synthetase, it can pick any amino acid. But to ensure that the amino acid attaches to its corresponding tRNA, and by corresponding I mean the tRNA carrying that anticodon, it must be selected. The selection is made possible because each one of those 20 amino acids has got its own amino acid tRNA synthetase. For example, in this case, we have got histidyl tRNA synthetase. For methionine, we're going to have methionine, methionine tRNA synthetase, meaning that the enzymes that select these amino acids are specific to that amino acid. And this specificity allows this amino acid to link to its corresponding tRNA. So what are we doing here? We want to ensure that we can interpret the message on the codon. The codon right now, as we speak, to just remind you, is CAU. We want histidine. How is histidine going to come to the ribosome? We understand it will be brought by the tRNA. But before it is brought the tRNA, something has to happen to the tRNA to make it suitable for it to come into the ribosome. And this is where we are. So this first step is charging the amino acid. Charging the amino acid or activating the amino acid is getting the amino acid that the codon is calling for and reacting it with ATP. This is a selection process, and it is made possible because the amino acid tRNA synthetase are specific to this amino acid. And it makes sense because we want only the amino acid histidine. So to select only histidine from the pool of those other 20 amino acids, we need its own amino acid tRNA synthetase. In this case, because the codon is histidine, the amino acid tRNA synthetase, which is the enzyme that's going to catalyze the reaction between your amino acid and your ATP, is histidyl tRNA. It means that if we look at the slide before, here, the amino acid tRNA synthetase should be methionine tRNA synthetase. It's the one that selected this um, methionine to come to the tRNA. So what this means is that each one of these codons, or each one of these amino acids, is going to have its own amino acid tRNA synthetase. So if you have got 20 amino acids, then how many amino acid tRNA synthetase do you have? At least there should be 20, because each one of them should be uniquely selected. So what you're going to have at the end of the day is the amino acid AMP. Of course, you're going to lose pyrophosphate. Okay. So this amino acid here has been activated. Now, after this activation step, this amino acid now is taken to its corresponding tRNA. So what you're seeing here is the tRNA. This tRNA must carry the corresponding anticodon for our histidine. That is the only way this histidine, which we activated in the previous slide, is going to bind to this tRNA. This amino acid AMP, we've activated it by attaching it to ATP. Now, to bind to tRNA, it won't just pick any tRNA in the cytoplasm. It can only covalently bond to the tRNA that is carrying the corresponding anticodon that is capable of complementary base pairing with the codon on the mRNA. Now, the process of covalently joining the amino acid which we activated to the tRNA is catalyzed by the same amino acid tRNA synthetase, meaning that these amino acid tRNA synthetases kind of serve two important functions. The first function is to select the amino acid and, of course, attach it to a high energy molecule, ATP. And then from there, when you form your amino acid AMP, the next step is to catalyze the covalent joining of this amino acid to its corresponding tRNA. And this is what you see here. So now, at this point, you have your charged tRNA. We are saying that it is charged because at this point, it is now attached to histidine. That is to say, because the codon we're trying to read is histidine. That is why I keep on referring to histidine. It can be any other codon. I'm just using histidine because in our case, the example given, the next codon we are reading is histidine. So we can say that this tRNA is now carrying histidine. How did this tRNA get charged? It was a two-step process. The first step is to fish out or select the amino acid. How was it selected? It was selected by a specific amino acid tRNA synthetase. In our case, it's going to be histidyl tRNA synthetase. Then from there, this amino acid now is covalently joined to the tRNA by the same enzyme. So at this point, you have your charged tRNA. This must happen to each tRNA that is going to bring in an amino acid to read a codon. So if the next codon is proline, again, you're going to have charging of proline from the cytoplasm. And then from there, you're going to have now attaching of that proline to the tRNA before this can go into the ribosome. And this process has to continue repeating itself. If the next codon is histidine, we need to charge another tRNA carrying histidine. If the next codon is methionine, we need to charge another tRNA carrying methionine. So the charging process just involves selecting the amino acid. Selection is based also on the specific enzyme. The enzyme is specific, meaning you have got 20 amino acid tRNA synthetases at least. Okay. Oh yeah, I think I should have brought this a little bit earlier. So this is just trying to say this tRNA must carry the anticodon that corresponds to the histidine that we are trying to read out. It is not an accident, or it is not arbitrarily, no. It is being charged with the amino acid that has been activated because it's carrying a corresponding anticodon that, when brought to the ribosome, can interpret this message as histidine. So on this end, it must carry histidine. Now, let's look at the genetic code, just a revision. We know that in the genetic code, we have 64 codons. And we know why we get 64 codons. We know that the language itself is 4 letters, right? And that language, we always put them in combinations of 3, which we're calling a codon. That is 4, and this N is the codon. We read them always in 3. So we've got 64 combinations that we can have, possible combinations. Out of these 64, we know that 61 can give us amino acids. Out of these 64, we know that 61 can give us amino acids. Okay, so these codons are also referred to as sense codons. Sense in the sense that they can give you an amino acid. And out of those 64, 3 of them are stop signals or stop codons, and we know them: UAA, UAG, and UGA. This is AUG is a start codon, isn't it? Of these 61, one of them is a start codon, AUG. Now, I want to find out if there are 61 sense codons, does a cell require 61 different tRNAs? Because at this point, we know that each one of these tRNAs must carry a specific, amino acid. And that amino acid is attached to that tRNA based on the fact that the enzyme will go in the cytoplasm, charge the amino acid, and then bring it to the tRNA. Now, we have got 61 codons. Does the cell need 61 tRNAs to ensure that all these codons are translated? I'm going to pause, and I want someone to attempt this question. I'll be sipping my tea as I wait. Remember, we are reviewing this topic. It's not like you're hearing it for the first time. So even some of this information, I'm sure was covered. But just in case you missed it, give it a try. Because at this point, the understanding here is we're just trying to remind ourselves that these tRNAs are being charged. The charging of a tRNA means it will be attached to an amino acid that corresponds to the anticodon it is carrying. We're saying it is attached to the tRNA with an anticodon that corresponds to it, in the sense that when this tRNA is brought to the ribosome, it's going to complementary base pair with the codon on the mRNA. Now here are the two things. The codon is on the mRNA, right? The anticodon is on the tRNA. We are saying the tRNA must carry an anticodon that corresponds to the codon. So let's not mix up the two. Now, going back to our question, we have got 61 codons, and we all agree because we've looked at the genetic code, guys. Now, if each codon must be translated, and we're talking about the 61, isn't it? Are we going to have 61 tRNAs?
No.
No, madam.
So, guys, I appreciate someone is giving it a try, and trust me, right now, no one is going to penalize you, and this is the time when you can make a mistake. So just give it a try and give me your reasoning to say, I'm saying yes because I'm saying yes, we're going to have 61 tRNAs, or I'm saying no, we won't have 61 tRNAs because, that's what I need. I want to understand your reasoning behind your yes or no.
Can I try, madam?
Of course, please give it a try.
I remember I was reading a book about that Francis Crick, if it's Crick, I'm not sure.
Francis
Yeah,
Crick.
yes, it stated that one single tRNA molecule can recognize and bind to multiple codons. So I think that's the reason why we don't necessarily need 61 different tRNAs.
Thank you for that. So now at this point, I want to understand. So after reading, did you believe what he wrote, or you think it makes sense? And if it makes sense, why? What's happening here?
Yeah.
Does anyone
Can
remember
I
one of the
can I add on
properties?
Adam?
Yes, please go ahead.
Because the third position on a codon can change. So I don't think we need 61 different tRNAs.
Thank you. So both answers are correct. Thank you very much, guys, for trying. That's very good. So here we need to understand. We're trying to charge this tRNA, and at this point, I hope everyone has understood. I've brought up this slide because I want us to have a comprehensive understanding. Yes, we understand we've got 61 codons. These 61 codons are also referred to as sense codons in the sense that they can give us a message for an amino acid. Now, if these tRNAs are going to be reading these 61 codons, do we need 61 tRNAs? The answer is no. And the reason is very simple. Like, our colleagues have said, one of them even read a book, which is very good because sometimes, you don't want to take what I'm saying, as gospel truth. I'm also a human being. I could actually even err in the way I'm explaining things. So it's good to always read widely to get the full picture. So remember during the the lessons or the lecture for the genetic code, we looked at a feature we refer to as the Wabo-based pairing. Wabo-based pairing, what does it mean? So when you look at, we're going to first of all look at the mRNA itself. I'll start with these two positions, these two codons. We've got this codon here, and then we've got that codon there. Both of these codons are supposed to give us an amino acid, phenylalanine. Okay? Both of these codons should give us phenylalanine. Please, you can verify. Maybe it is even wrong. It can be anything. But what we're trying to say is that this codon here is going to specify phenylalanine. This codon here is going to specify phenylalanine. Now, let's look at this codon. If you look at the first codon, reading all the time from the 5' end to the 3' end, please, all the time, we are reading the mRNA from the 5' to the 3'. So I'll start at the 5'. We have UUC. And remember, in the previous lectures on the genetic code, we even gave these positions, isn't it? This is the first position of the codon, second position of the codon, and that's the third position. So now let's look at the next codon that we're saying the information it's carrying is also supposed to be translated to phenylalanine. We'll start from the first position of this codon, reading from the 5' end. At the first position, it's a U. When you compare this first position and that first position, they're the same. Let's go to the next position of this codon. There's a U here. Let's compare this to the other codon. There's also a U. Let's go to the third position of this codon. It's a U. The third position here is a C. What we get from these two codons that both translate or give us information for phenylalanine is that the first two positions match, but the third one can allow for a mismatch. And this is what we're referring to as Wabo-based pairing. It's flexible. So meaning that even the tRNA that is going to read these codons is going to be the same, but to also exhibit the feature of Wabo-based pairing in the sense that this position here can allow for a mismatch. Let me say that again. So here we're looking at the feature of the genetic code referred to as Wabo-based pairing. In Wabo-based pairing, the first two positions in the codon, reading from the 5' end all the way to the 3' end, all the time is completely spare. It can allow any mismatch. Okay, the tRNA that is going to be the ribosome must be the tRNA that is going to be changed by the methionine. You can understand, isn't it? But what should it carry for anticodon? Remember, the relationship between the tRNA and the methionine is made possible by the anticodon-codon interaction. Now, it means that on the third position of this tRNA, it can also allow for mismatch. So you can see there's AA, there's G. G pairs with C. So this is complementary base pairing. But when it comes to the anticodon, you can see that G just base pairs with U. This is a mismatch, but this mismatch is only at the third position. Meaning that when looking at these codons in the genetic code, the first two positions are very, very accurate reading. The third position can allow a mismatch. Now, let's not take this for granted. it doesn't mean that now you can best pair anything mismatched. You can that not best pair with codon 4 leucine is UUA. You can see the position of here. Okay, so it can mismatch. It can rule that it has to. It always get A and A. It's never be a G like in this case. For example, this is also leucine. The codon is UUA. UUA is UG message for are the same. The position is different. But because this TRNA is for, in fact, some of it are not based with an adenine with a guanine. In fact, have a G and it's to be too bulky. Okay. So what I'm trying to say here is that we have got 61 codons. Yes. And we know how we get those 61 codons. And those codons have to be interpreted by a tRNA bringing an amino acid to the ribosome and interpreting the message on our codon. But because of Wabo-based pairing, we don't need 61 tRNAs. No, they are less than that. And the reason is because the third position on the mRNA, or indeed in this case, even on the tRNA, can allow for a mismatch. This position is Wabo base. This is a feature or characteristic referred to as Wabo base pairing. I put this slide just to remind ourselves to say, yes, we are charging our tRNA. Charging the tRNA just means the amino acid in the cytoplasm is selected by a specific enzyme, meaning that we only need 20 amino acid tRNA synthetases because we just have 20 amino acids that exist naturally, isn't it? So many, but then for the tRNAs, we don't need 61 to interpret the codons. Why? Because of this feature of Wabo base pairing. What about the stop codons? Do stop codons have their own tRNAs? Right now, remember we said we have got 61 tRNA, 64 codons, isn't it? We've looked at the 61, the sense codon. The stop codons are sometimes referred to as nonsense or nonsense codons because they don't give us any amino acid. But I want to find out, do the stop codons have their own tRNAs? And then, before you answer this question, I want you to just think a little bit more. What is the tRNA doing? Because your answer will even indicate to me whether you've understood what amino acid activation or tRNA charging means. This question, I want us to understand, to ask ourselves, the stop codons. The stop codons, those are signals in the genetic code that tell us where to stop. If it is in a sentence, if you are reading a sentence, when you reach a full stop, you stop, isn't it? So meaning that dot, the full stop itself, tells you the message has ended. So you can think of a stop codon in the same way. But the question here is, do the three stop codons have their own tRNAs? If the answer is yes, please tell me why. If the answer is no, please tell me why. I'm going to be sipping my tea as you guys, think through that question.
Madam, can I try?
Of course, anyone, you guys, you're free to try. Try and make as many mistakes as you can. Remember, it's not an exam.
Yeah. I think no, because the stop codons do not even require any tRNA. In fact, there are no tRNAs to even recognize stop codons unless start codons. So I like to say no, because the tRNAs carry amino acids, and the stop codons do not code for any amino acids. So the best explanation I can put this is that no, stop codons do not require their own tRNAs because there are no any tRNAs to exist any stop codon. Yeah.
Thank you. Thank you. I'm not saying, thank you very much for that answer. And at this point, I'm not saying it is wrong. I'm not saying it's correct. Does someone else have a different answer to that? We all agree with what was talking, Aline Nani. We all agree. Or maybe we didn't.
Yeah,
Yes, we
We
agree.
agree.
I agree.
Okay, good. So what Alinani was saying here is that the stop codons in the first place, they are just a signal to ensure that the message ends here, isn't it? Translation must stop. And we even said we can think of a stop codon like a full stop. Because I think when we're reading our sentence, when we get to a full stop, we even slow down, isn't it? Before you start the next one. But at least you know this sentence ended here. In the same way, the mRNA carrying information for making proteins has got a full stop, or should I say stop codons. Now, the stop codons do not carry information for any amino acid. They don't carry any information for any amino acid. Of the 20 amino acids, they don't carry any information for any amino acid. Now, if you're going to ask the question, do stop codons have their own tRNA? You must ask yourself, what is the tRNA doing in this picture, in translation? The tRNA is like an adapter, right? The tRNA is an adapter. It is carrying on one end the acceptor arm. On the acceptor arm that is found on the 3' end, we know this is where the amino acid is going to be attached, isn't it? That amino acid that's going to be attached to tRNA corresponds to what the anticodon on that tRNA is. And by corresponding, we're saying that the anticodon is going to complementary base pair with the codon on the mRNA, carrying information for an amino acid. So if the codon on the mRNA is not carrying any information for an amino acid, what will the tRNA be doing in that picture? What is it going to be carrying on the acceptor arm? So thank you very much for those answers. But I just wanted us to understand, to just and to appreciate. I wanted to appreciate whether you've understood what tRNA charging is. tRNA charging, you want to interpret what is sitting on the codon. If that codon is a sense codon, it means it's going to give you an mRNA. So it's going to give you an amino acid. How is that amino acid going to be brought to the ribosome? It's going to be brought in by an adapter we're calling tRNA. On one end, the tRNA will interact with the codon. So we're going to have anticodon-codon interaction. And on the other end, it must carry the corresponding amino acid. If it is a stop codon, the tRNA has got no space in that picture. What is it going to be doing? What is it going to be carrying on the acceptor arm? And this codon-anticodon relationship, how are we going to establish it? Because remember, there's no information for a particular mRNA. In fact, these anticodons, these stop codons are recognized by release factors, and that is something that we covered in termination. So this is just a slide on stop codons, but the take-home message here is that stop codons do not give us information for amino acids. And because of that, and we know the function of the tRNAs are adapters. They go in the cytoplasm, get activated by binding to an amino acid, and then bring that amino acid to the ribosome and establish a codon-anticodon relationship. And then you can have your translation. Okay. So now, at this point, we are done with one part, which is how the tRNA is functioning as an adapter, or just the role of tRNA in translation. Guys, you can imagine we haven't even started translation initiation, but you know what? It has been the lecture has been put in this way so that we just understand the components that we need. We need to bring in these amino acids into the ribosome. But how is this amino acid coming to the ribosome? We need to understand the process of just charging the tRNA itself. And I hope at this point it is clear. So now, the next thing that we want to look at is to answer this question. How does a ribosome ensure that the correct charged tRNA enters during translation? Now, I'm going to take you to this slide. Remember, we said that the ribosome itself has got 3 compartments. Okay, it's got an A site, amino acid site, peptidase site, and and the exit site. We know that the amino acid site, so it is just A, P, E. These are the sites in the ribosome. I want us to understand that these are like compartments in our house. Okay, we have a kitchen, we've got a dining room, living room, and bed. All of them have got different functions, isn't it? I don't think we are sleeping in the kitchen. We call it a kitchen because that is where we're supposed to cook. And then we call a different space a bedroom because that is where we're supposed to sleep. So when you think of a ribosome, it has got these different compartments. In one compartment, the A site, this is where your charged tRNA, and at this point we know what charged tRNA is brought into the ribosome to read the message that is sitting in the A site. The A site, the amino acid site, this is the entry point for the charged tRNAs. The only tRNA that is going to come into the A site and complementary base pair with this codon in the A site is the one that is capable of forming a stable complementary codon anticodon relationship. We are saying that the ribosome has got compartments. We can think of them as compartments for now. One of the compartments is the A site. What does the A site do? The A site is like the receiving bay. It receives charged tRNAs. For example, in this slide, what we can see, we've got the charged tRNA, and at this point we know how this guy got there. The anticodon on this tRNA, which is AG, must correspond to the codon sitting in the A site. Remember, this is the mRNA, and right now we have got this codon in the A site. This codon is UUC. And this codon should be translated to phenylalanine. That is why only the tRNA carrying phenylalanine can be allowed to come into the A site and form complementary base pair with this codon. What about the P site? I'm moving in this direction and we're going to see in the next slide or so how this is happening. I just want us to understand at this point, we've got the A site. The A site is the receiving bay. If you own a shop or if you've been to ShopRite, you know they've got those, receiving bays at the back of these shops that where they get a lot of stuff and then it is sorted. So think of the A site as the entry point of charged tRNAs. These charged tRNAs don't just come in here and bind. No, there's a rule here. Remember, we need to have a stable anticodon-codon interaction. Meaning that if at this position of the A site, and remember it has to be a complete codon. In the A site, we must have a complete codon sitting there. In this case, for example, we've got UUC. This is a complete codon. It can give you an amino acid. Only the tRNA that is charged with phenylalanine can come and complementary base pair here. So this is like a receiving bay. What about the P site, which is also referred to as a peptidosite? In the P site, this is where the peptide bond is formed, where the amino acid that is sitting in the P site, we'll talk about that, forms a peptide bond with the amino acid that is going to sit in the P site, in the A site. The peptidosite, this is the site where the peptide bond is going to be formed. The peptide bond will be formed by the amino acid that is going to be present in the peptidosite, in the P site, with the amino acid that's going to be present in the A site. We'll look at that in a short while, because at this point now you're wondering, if the A site is a receiving bay, this is the entry point, how did this tRNA get to that position? We're going to see that in a bit. But for now, I want us to understand that the amino acid that sits in the P site, of course, we know this is the tRNA is going to form complementary base pairing with this codon on the mRNA. Now, the amino acid that is bound to this tRNA is going to be transferred to the amino acid that's going to sit in the tRNA, in the A site, by the formation of a peptide bond. We know that amino acids are linked by peptide bonds. The moment this tRNA transfers its amino acid to the one sitting in the A site, it loses its stability for binding and it enters into the exit site and leaves uncharged. What you see here, for example, is a tRNA that is no longer carrying any amino acid. What has happened to this amino acid it was carrying? When it was in the P site, this amino acid was transferred to the amino acid that was sitting in the P site. So anyway, so this is the entry point, like a bay, the receiving bay. This is the point where the tRNA forms complementary base pairing with a codon, but is carrying an amino acid which forms a peptide bond with amino acid in the A site. Once the peptide bond is formed, amino acid is cleaved from the tRNA. The tRNA at this point is no longer stable to bind to the ribosome and the mRNA. In fact, what you see here is that one of the things that enable translation is that the tRNA, when it is charged, it has got a high affinity for the mRNA and also a stability in the ribosome. So once it loses its amino acid, it's no longer stable. It moves to the exit site and it gets out of the ribosome. So this is the exit site. In the exit site, the tRNAs exit without an amino acid. This amino acid was transferred in during the time when this tRNA was in the P site. So let's look at translation initiation. I'm going to explain that step by step. Okay. So let's start with translation initiation. Okay. So what is translation initiation? When you think of translation initiation, this is a step, the first step of translation, where a tRNA is going to be brought in the A site. In addition to that, it is also a step when the ribosome associates with the mRNA. The association of the ribosome with the mRNA should allow for the start codon to be positioned in the P site. Translation initiation as a whole, let's just look at it. We need that the ribosome identifies or associates with the mRNA, because we are saying that the information on the mRNA, the codons, are going to be translated in the ribosome. So meaning that this ribosome has to associate with the mRNA. Remember, these ribosomes translate a lot of RNAs, so they're not permanently bound to these mRNAs, no. In fact, we're going to see in a few slides that even the binding of the association of the ribosome and the mRNA is a sequential process. But in this slide, we're just trying to understand what is translation initiation. It is just a process where the ribosome associates with the mRNA. This association must allow for the start codon, the first codon that is supposed to be read on this message, to be positioned in the P site. Remember what I said? This A site is like a receiving bay. All other tRNAs that are going to read any codons that come after the AUG come in the A site. But this is a unique case. To start initiation, the first tRNA comes in the P site. Let me say that again. Remember in the previous slide, I said the A site is a receiving bay for all tRNAs that are supposed to read the codons that sit in the A site. But the first tRNA that comes into the ribosome does not come in the A site. It comes in the P site. So meaning that to initiate translation, the ribosome must ensure that the AUG site codon is positioned in the P site and not anywhere else. And this step is sequential. And what you see in this slide is formyl N-formyl methionine. Now, this is F-met. What this means is that this tRNA is being activated with the methionine. Why? Because we know AUG, the start codon, the message here should be translated to methionine, isn't it? But one thing you're going to note is there's an F there, and I'll explain why this is so. This tRNA is unique in the sense that it does not go to the A site. It goes straight into the P site. So we can also refer to as the initiator tRNA. The initiator tRNA is unique in the sense that it does not enter the usual door for delivery, which is the A site. It goes into the P site. In addition to that, you're also going to see that instead of it just being just tRNA methionine, it carries a modification formyl group, N-formyl methionine. So we're going to see why this is different. But the take-home message from this slide is that to initiate translation, we need the ribosome to associate with the mRNA. This association should facilitate the positioning of the AUG, which at this point we know is a start codon, in the P site. This codon should not be in the E site, it should not be in the A site, it must be in the P site. This ensures that the tRNA that is charged in the cytoplasm should also just come in the P side to translate it as methionine. Now, where is the problem going to be here? The mRNA also carries other AUG, codons. What I want us to understand here is that the AUG is a start codon, right? And it gives us the message methionine, amino acid methionine. But that doesn't mean that AUG at the beginning is the only AUG that an mRNA can have. Yes, some of them only have one. But there are times when a protein has got more than one amino acid methionine. For example, in this case, we have got methionine here. AUG should give us methionine. There's this codon, and somewhere else along downstream, you have another methionine. So we can regard this one at the beginning as a start codon. We can regard this one as an internal codon. Now, how does the ribosome ensure that it positions this one and not that one in the P site? Remember in the previous slide, what am I saying? To start initiation, you need this AUG to be positioned in the P site. And this should be the start of the message. So you should think of it, if this were a sentence, if you start reading this, if this is the AUG positioned in the P site, it means all these other codons will not be translated. The message is going to be truncated, like abrupted, isn't it? If you're going to write a sentence, it must have all the words that you want to put in that sentence to have the full meaning. So then, how does the ribosome ensure that it can, separate this start codon that indicates where the message starts from any other codon that can be downstream that can also carry methionine? This is to understand that when we say AUG is a start codon, this does not mean that it is the only codon that can exist as AUG. Internally, you can have other codons that also give out the message methionine. So then, how does the cell at initiation, the ribosome, ensure that it can only position this one and not any other AUG in the sequence in the P site? The first thing we need to understand here is that the ribosome ensures that by reading the mRNA only from the 5' end, scanning it from this end to that end. That is to say, reading has a sequence. We remember when we're looking at DNA replication, transcription, and so we say, the process is taking place from the 5' end to the 3' end. So one of the ways that the ribosome ensures that it does not make a mistake to position a different AUG in the P site, and only the one that's at the beginning in the P site, is that it starts reading the mRNA from the 5' end sequentially. And sequential, I mean codon by codon. Okay? So that the moment it comes to AUG, it knows this is the first one that because it will be scanning as it moves from this end to this end. So it will see this is not AUG, so it won't position it in the P site. It will go to this one, this is not AUG, I shouldn't position it. Then it finds, okay, this is now AUG. This is the guy that should be in the P site. And then the other ones will follow. So meaning that there's no chance of this guy finding itself in the P site. So this is the take-home message from here. Now, in addition to just having a specific direction to read the mRNA, the mRNA itself has a sequence that is referred to as a Shine-Dangano sequence, this one here. What I'm trying to say here is that we agree that one of the ways that the ribosome ensures that only the first AUG is positioned in the P site is just by reading it sequentially from the 5' end to the 3' end. In addition to that, the mRNA itself also carries a signal that is referred to as the Shine-Dangano sequence. Guys, my pronunciation could be wrong, but at least I hope you're going to understand what I mean. We're trying to establish how does the ribosome ensure that only the first AUG is placed in the P site. It should not read any other one. First of all, it's just a sequence of reading. Like us, if I give you something to read, there's no way you're going to start reading from the left, from the right to the left. We read all the time from the left to the right, isn't it? In a similar way, the ribosome also reads from the 5' end to the 3'. That's a sequence of reading. That's the first thing it does. The second thing, it gets a signal in the mRNA that is referred to as a Shine-Dangano sequence. This Shine-Dangano sequence is found upstream of the AUG. So what we see here, this is AUG. AUG, this is going to be the first AUG. Before the ribosome even interacts with the AUG, it will also recognize this sequence, which is referred to as a Shine-Dangano sequence. This sequence is on your mRNA. The Shine-Dangano sequence is sitting on the mRNA. The ribosome has a binding site for this sequence. That's the only way it can recognize it. Okay, the ribosome will start scanning from the 5' end. As it scans, it's going to find this sequence. The Shine-Dangano sequence has a binding site in the ribosome, in the small ribosomal unit. Okay, so this complementary base pairing between the Shine-Dangano sequence of the mRNA and the ribosome also facilitates the positioning of the AUG site in the P site. So now we are saying that there is no confusion here. I'm going to go back here. The ribosome will not position any other AUG in the P site. It's going to use one, the sequence of reading from the 5' end to the 3' end to ensure that it can read this codon and position it in the P site. In addition to that, before the ribosome even gets to this AUG site, it also has a sequence upstream. And we know what upstream means this time around. If this is my AUG, and I'm saying the Shine-Dangano sequence is upstream, I'm referring to sequences going this side. If I'm talking about sequences downstream, the AUG, I'm talking about sequences going this side. We're saying that the Shine-Dangano sequence is on the mRNA upstream of the AUG, approximately about 6 or so nucleotides away from the AUG. So these two things allow for this AUG to be positioned in the P site. Okay, so now what does it mean? If you have an AUG, that is, at this point, we're referring to it as a start codon, and we know it's a start codon. The other AUGs, we can also, that are found internally of the mRNA, we can call them as internal codons. Okay, these are not start codons anymore, because remember, an mRNA can only have one start codon, this one. So meaning that the other AUG, if they exist in that mRNA, we refer to them as internal codons, and they'll be treated just like any other codons. So then here there's a challenge. The codon that must recognize this guy must be different from this one that should treat this guy as an ordinary codon, because this codon is quite unique. It is unique in the sense that it marks the beginning of a message. This codon here is not a marker of the beginning of a message, it just specifies methionine. Yes, both of them give us methionine, but the one at the beginning has a unique feature. It is the one that indicates where the message starts. This guy here, it also gives us methionine, but there's really nothing unique about it. It's as good as the other codon that gives us something else. So the cell needs to treat these two codons differently, despite them giving us methionine. What that means is that every cell has got at least two tRNAs. The tRNA that should recognize the first one, which is AUG, the start codon, and the tRNA that should recognize an internal AUG, which I'm just going to refer to as internal codon. At this point, I don't want to confuse you. I just want you to understand to say, when we have AUG as the start codon, we're not trying to say the mRNA cannot have another AUG. It can have multiple, 5, 6, 20 of them. But the ribosome has to distinguish which one of those is the start of the message. One of the things that it also does, apart from just a ribosome reading and the Shine-Dangano sequence, is also the tRNA that comes to read them. They are unique. The tRNA that reads this first codon is modified. In bacteria, for example, it carries a formyl group, N-formyl methionine tRNA. This tRNA is just going to be, it will not be modified. Let me use this slide. So here, regard this one as the start codon, AUG. And then this one, we're going to regard it as an internal AUG. That is to say, on the mRNA, the AUG, this one is marking the beginning of the mRNA message. This one is just found internally in the sequence of the mRNA. Now, this is not always the case. We're just trying to show that when there's an AUG internally of the mRNA, it will be recognized by a different tRNA, different from the one that reads the start codon. The tRNA that reads the first codon of the mRNA or the start codon is referred to as the initiator tRNA. The tRNA that reads the first codon, the start codon, the one that goes in the P site after it's been positioned by the Shine-Dangano sequence, is referred to as the initiator tRNA. And this tRNA is modified. That is why you see there's an F. It is formulated. I might not pronounce it very well. But this tRNA that reads the AUG away from the start codon is just referred to as the elongator tRNA. So this is AUG. This tRNA is going to be referred to as the elongator tRNA. Okay, these two tRNAs are different. Initiator tRNA reads the first AUG. Elongator tRNA then reads the first AUG. It reads any AUG internally of the mRNA sequence. So please distinguish that. Because we are looking at bacteria, that is why we're saying it is formylated. Yeah. So let's look at now what we need to initiate translation initiation. So we're clear at this point, we need the mRNA. We need the 30S ribosomal unit, because remember I said that the mRNA is not sitting permanently in your ribosome, because the ribosome, if your mRNA sits permanently in the ribosome, how is it going to translate other mRNAs? So it means that the ribosome has to be assembled. The assembly of the ribosome means you're going to have a 30S ribosomal unit alone, 50S ribosomal unit, and then you assemble the whole ribosome. So that is why the 30 ribosomal unit is shown independently. You're also going to need your initiator tRNA. Initiator tRNA, like I said, it is referred to as N-formylmethane. It is modified. In short, it's just referred to as FMET or FMET tRNA. That FMET means this is initiator tRNA. A tRNA that recognizes internal codon does not have an F. It will just be MET tRNA. It will not have the F at the beginning. We also need initiation factors. This is something new. Remember what is on this slide, we've already covered it. We know we need the mRNA. It's going to carry that information that is need to be translated. The smallest ribosomal unit, it is just a member of the ribosome. We have the large subunit and the small subunit. Initiator tRNA, we know why we need this one. We need this guy because we need to interpret the first AUG, which gives us methionine. And because of that, it's a first one. This methionine goes in the P site. This guy is modified in bacteria with a formyl group. So it's referred to as N-formylmethionine, short just FMET. Okay, then we also need something that is referred to as initiation factors. These are proteins, okay? These are proteins that help the ribosome itself to be, the mRNA to be positioned in the mRNA to position in the ribosome. It also prevents premature association of the 30S ribosomal unit to the 50S. Like I said, the ribosome does not permanently bind to these mRNAs. It can only associate with one that needs to be translated. So meaning that at some point, they are independent. So now, the ribosome, before it can allow for the mRNA to bind, it must not associate with the 50S ribosomal unit to form a complex. So some factors have to come in to stabilize the 30S ribosomal unit, allow for the mRNA to bind, then the 50S ribosomal unit can come and bind. This process also is an energy consuming process. Translation is an energy consuming process. And remember, I always tell you in class to say, let's conserve energy. And you guys, I even encourage you to eat. Spend your energy very well, because our bodies need quite a lot of energy. And you can already see, to just get a protein, we need to spend a lot of energy. Remember, during the activation of your tRNA, you used a lot of ATP. This time around, to get your protein initiation, you use GTP. This is quite a lot of energy. So let's think very well how we, expend our energy resource in our systems. Then you also need the 50S ribosomal unit, because we know that the ribosome functions as a unit. It's referred to as 70S, but we know the two subunits is the 30S ribosomal unit and the 50S ribosomal unit. We are saying 50S ribosomal unit independently in this case because we know at the beginning we don't have the whole ribosomal unit assembled. We need to give chance for the mRNA to slot itself in the ribosome and then bring in the other big guy. Then we also need magnesium ions. These magnesium ions, they help stabilize the ribosomal structure or just maintain the relationship between the mRNA that is going to be read and the tRNA that is going to be coming in to bring in those ribosomes. So we need quite a lot of things. Now, I'm not trying to say you should memorize these things. No, there's really no need for us, but I hope you can just follow what we're trying to say here. For example, we need the ribosome. The ribosome comes in as a 30S, And the 50S, we're looking at bacteria, isn't it? It's not a eukaryote. In addition to that, we need energy. The energy that you're going to use in initiation is GTP, different from what you use when you're activating your tRNAs. That's why you use ATP. In addition to that, we also need initiation factors. What you're seeing here is IF1. IF is initiation factor, so I'll just refer to it as IF. This is IF1, 2, and 3. All of these you're going to see, they play different roles. And I hope that as I go through the slides, you're going to capture all these things that we're going to need. The mRNA itself, because we need someone to give us information that we're going to translate. The TS ribosomal unit, initiated tRNAs. Of course, we need other tRNAs to come in as well. So let's look at what happens when assembling this ribosome. So formation of the 70S ribosomal initiation complex. At this point, are we following? Before I even, I've been talking a lot. So I just want to understand whether people are even following or you're just confused, because I don't have, a review of this lecture again. Are we following up to this point?
Yes.
Are there any questions up to this point? Have I lost someone? Because sometimes when you explain, to me, it might sound, I'm not too sure. Someone is saying we should turn off. Who should turn off their microphone? Should I turn off my microphone? You want to say something? Okay, what I'm trying to ask here is that I want us to follow. We're remaining with just a few slides, we'll be done. But I want to ensure that up to this point, we have followed each other. We want to form the 70S ribosomal initiation complex, meaning that we're still in initiation. And at this point, before you even initiate, we understand our tRNAs also need to be activated, and we know how that is done. Now, when we say we want to form the 70S ribosomal initiation complex, we understand the 70S consists of two parts. It has got a 30S ribosomal unit, which we refer to as a small subunit, and then it's got a 50S ribosomal unit, which is the large subunit. So together we know we form the 70S ribosomal unit. Guys, there's someone with a microphone on. Thank you. So what I'm trying to say here is that when we are saying we want to make a 70S ribosomal unit, the understanding right at the beginning should be that the 70S ribosomal unit consists of two parts. It consists of the small ribosomal unit, which is the 30S, and the large subunit, which is the 50S. Together, when they form a complex, they form a 70S ribosomal unit. Now, we are saying we want to assemble this 70S ribosomal unit because remember, it does not bind ribosome mRNAs and stay permanently bound to them. It only assembles on an mRNA that needs to be translated, meaning that at the beginning, they must occur as independent subunits. By independent, I mean the 30 ribosomal unit to be on its own, the 70S ribosomal unit also be on its own. It is only when it is presented with an mRNA can these two come together. And before they come together, they must allow for that mRNA to slot itself or associate with the ribosome. The first point of association of the message on the mRNA with the ribosome is a small ribosomal unit. Of the two ribosomal units, the small ribosomal unit and the large ribosomal unit. The first thing is that the ribosome has to associate with the mRNA. Which of these two subunits first associates with the messenger RNA? It is a small ribosomal unit, the 30S. So now let's look at how we're going to assemble this. So the first thing we need to do is to form a 30S pre-initiation complex. Why? Because I said the ribosome has got two subunits, isn't it? One of them has to first of all associate with the mRNA. And this one of the two ribosomal units is a 30S ribosomal unit. That is why we're trying to say formation of the 30S pre-initiation complex. Pre in the sense that this is the 70S, meaning it's complete. But the one that is formed before is like a pre, like a smaller, like an initial step in formation of the larger complex. So formation of the 30S ribosomal unit is just a process where you have the small ribosomal unit interacting with the mRNA. This interaction, I've already mentioned, involves this small ribosomal unit recognizing the Shine-Dangano sequence. How does it recognize it? It has a binding site in itself that can recognize that subunit. And this allows the mRNA to be positioned in the P site. So remember what I explained earlier, that the ribosome reads this mRNA in a sequence from the 5' end of the mRNA to the 3' end, meaning that the ribosome is going to be moving from the 5' end of the mRNA going towards the 3' end of the ribosome of the mRNA. Now, the first step is to associate the mRNA that needs to be translated with the ribosome. This association is first with a small subunit. And this association involves what? It involves the 30S ribosomal unit recognizing the Shine-Dangano sequence. Or should I say the first thing is that the ribosome should read this mRNA from the 5' end to the 3'. That's how it should be translocate moving. In addition to that, it must recognize and bind the Shine-Dangano sequence. In addition to that, it must, after recognizing the Shine-Dangano sequence, position the AUG in the P site. This must happen at the beginning while we form our 30S pre-initiation complex. The 30 is coming from the soft small ribosome unit. What do we need? This process needs initiation factors. Okay, for example, we need IF1 and IF3 to stabilize the ribosome. You're going to see that IF1 sits in the A site, blocking it, okay, so that the first tRNA does not go in the A site. Remember, I said the A site is like the receiving bay, meaning that every tRNA that should come into the ribosome to read the codon must first enter the A site. But in this unique scenario, the A site is blocked. This blocking of the A site ensures that the first tRNA, which is the forming methionine in bacteria, goes to the P site. So we need initiation factors. And like I said also, we need to recognize the Shine-Dangano sequence. And of course, the tRNAs and so on and so forth. Let's look at a more visual picture. So let's imagine this as the 30. This diagram here is showing what is happening during the formation of the 30S pre-initiation complex. So this is a small subunit, okay, small ribosomal subunit. It has got an A site, a P site, of course, and an exit site. For demonstration, let's just keep it like, okay, we haven't shown the E site, but it's still there. So the first thing that the ribosome needs to do is to stabilize itself. By stabilizing itself, I mean it must not associate with the 50S ribosomal unit before the mRNA comes in. Like I said, these two subunits occur independently before reading the mRNA. So this ribosomal unit must ensure that it is independent of the 50S, does not prematurely associate with the 50S ribosomal unit. How does it do that? The IF3 comes in and binds the ribosome, making the binding associated with the 50S unstable, so that it remains like a single unit. In addition to that, we want to ensure that the tRNA that's going to come in the ribosome when translation begins does not enter the A site. What happens here is that the IF1 sits or blocks the A site. In addition to that, you're going to see that you're also going to bring in the mRNA. The mRNA is brought into the ribosome, and the positioning of the AUG is facilitated by the reading of the mRNA from the 5' to the 3', in addition to recognition of the Shine-Dangano sequence. This is what you're seeing here. So our A site is blocked. The bay for receiving tRNAs is blocked. So meaning that even if there's any codon there to give us an amino acid, there's no tRNA that can come and read that codon. The first codon has been positioned in the P site by the reading frame and the Shine-Dangano. This is what is normally happening when forming the 30S pre-initiation complex. Now, how do we bring in the tRNA into the ribosome? Let's look at the next slide. I hope this is clear. The first thing is that the mRNA must associate with the 30S ribosomal unit. This association is made possible because this unit stays independent of the 50S, does not associate prematurely. This premature association of the 30S ribosomal unit to the 50S is prevented by initiation factor 3. It binds to the small ribosome, making it unstable and favorable to bind. In addition to that, the A site must be blocked. The S site needs to be blocked because remember, the first codon that's supposed to be read is not placed in the S site. It is placed in the P site. How is it placed in the P site? The mRNA itself carries a signal we're referring to as a Shine-Dangano sequence, which the ribosome is going to recognize. The Shine-Dangano sequence is not recognized by the 50S ribosomal unit, it is first recognized by the 30S ribosomal unit because this small ribosomal unit has a binding site for the Shine-Dangano sequence, and this binding allows AUG site to be fixed in the P site. Now, keep in mind, while the A site is blocked, it still carries a codon, right? Because remember, after the start codon, there's still a message that goes on this side. Only that we can't read that message because the A site cannot allow any tRNA to interpret that codon. But the P site is positioned, sorry, the AUG is positioned in the P site. Okay, now, how do we recruit the initiator tRNA? Remember from the previous slide, we said the initiator tRNA is a tRNA that reads the first codon of the message on the mRNA. And we know the first codon is a start codon. So this initiated tRNA has to be brought to the ribosome. The bringing of this initiated tRNA to the ribosome is facilitated by a complex of initiation factor 2 and GTP. It's an energy-dependent process. What I'm saying here is that now we have our AAG positioned in the P site. In addition to that, we also have our 30S ribosomal unit independent of the 50S because IF3 made it unstable. This tRNA cannot make a mistake and go to the A site because the A site is a receiving bay, isn't it? Because the A site has been blocked. So the only place where it can go and stably bind is the AUG. You may wonder if the E site is still there, why doesn't it go in the A site? Remember, the stable interaction between the tRNA anticodon and mRNA codon is based on complementary base pairing. Now, this tRNA is carrying an anticodon that can only complementary base pair with this AUG. So even if it went in the E site, it cannot stay there. It can only sit where there's going to be stable codon-anticodon relationship. And this one ensures that this situation only occurs in the P site, right? So I hope we've understood here. This tRNA that is going to be brought to interact with this codon, remember what happened to it. It can only have stable binding if it has been charged. So that is why it was important for us to understand the charging of this tRNA. This tRNA has to go into the ribosome, select the amino acid methionine because this codon here is calling for methionine. Once that amino acid is attached to the tRNA, then that tRNA now can be brought to the ribosome. The tRNA doesn't just come to the ribosome on its own, it is facilitated, or should I say helped, by who? By an initiation factor. Which one of those three? Remember I gave you three, IF1, 2, and 3. IF3 is already sitting on 30S. IF1 is blocking the S site. So of those three, the one that comes in to bring this tRNA, charged tRNA by the way, is IF2. IF2 does not come alone as well. It forms a complex with a GTP. So this is going to be like a complex. You're going to have FMET, or should I say IF2 GTP FMET complex. Then these three are going to move to your ribosome, like what you can see here. You've got your tRNA, you've got your initiation factor, and the complex with GTP. What happened here? This tRNA was helped to come to this position in the P site by forming a complex with the initiation factors. So initiation factors, they play a number of roles. One of them is just to stabilize the ribosome to prevent premature association. Another one of them is to block the A site so that if you're going to allow any tRNA to come into the ribosome, it can be fixed in a particular position. The desirable one initiation is the P site. And the tRNA that's going to come in the P site is going to be enabled to come through by a formation of a complex of initiation factor 2 GTP with that tRNA. Then together they're going to come and sit in the P site. This is made possible because of the anticodon-codon relationship. It must be complementary. Is that the case? Yes, this is AUG. On the tRNA, it must carry complementary anticodon. You can see this is A base pairs with a U because this is RNA, not DNA. Then U should base pair with A and G with a C. So there's complementary base pairing. So at this point, we are done with assembly of what? We're done with assembly of the 30S pre-initiation complex. The next thing is that the ribosome does not function as only the 30S. It functions as a whole. It must be a 70S ribosomal complex because the 50S has to come in and bind. Now remember, the 30S ribosomal unit had this IF3. IF3 was making sure that the 50S does not come in so that it can allow for this to take place. The IF3 was ensuring that this guy is so unstable to bind the 50S. But when this has been established, at least we have our tRNA sitting in the P site. How does it ensure that there's complementary base pairing? This site here is blocked. Now we can bring in the 50S ribosomal unit to mix the whole complex because the ribosome to translate needs the 50S and the small s ribosomal unit. So how does it happen? So the 50S ribosomal unit itself, when this initiator tRNA binds to the codon in the P site, it causes the complementary base pairing causes a constraint, or should I say, it allows for the GTP to dissociate and other insertion factors to come off from the ribosome. And what I'm trying to say here is that the stable binding of this tRNA to this codon in the P site, it causes these insertion factors to dissociate. Okay, it's like the binding pattern has been established. At this point, we no longer need the A site to be blocked because we have now established the first position of the tRNA to sit. And also, this ribosomal unit can now bind the 50S ribosomal unit because now we've established the reading frame. That this is where we're going to start. How has that been established? Complementary binding of FMET, recognizing the first codon in the P site. Now, the dissociation of these initiation factors is an energy consuming process. Remember, we've got GTP. So GTP is hydrolyzed, getting rid of all these guys. We don't need IF3 anymore because we need this guy to now associate. We don't need the A site to bind, to be blocked, because at this position, we've established the first codon. The tRNA is already sitting there. Now, when this happens, all these guys have left. We don't need them anymore. This is now the formation of the 70S initiation complex. At this point now, we are ready to initiate translation. But as you can see, it is a stepwise process. You activate your amino acids. Your small ribosomal unit has to be alone. You shouldn't bind to the 50S. The S site must be blocked to ensure that the first tRNA comes only in the P site. When that tRNA comes into the P site, it ensures stable codon-anticodon. This establishment of this stable codon-anticodon relationship, it allows for the other IF factors to dissociate. You don't need the A site to be blocked anymore because now you can allow anyone to come in to bind to the 50S. We're done with initiation. Are there any questions? A long time. I hope initiation of translation. Just if there are no questions, guys, let me proceed to elongation. So what does elongation mean? Just by looking at what we can see here. Elongation means that we're supposed to bring in a tRNA to read this codon. Like I said, in the S site, the codon must be fixed, 3 nucleotides, like 1, 2, 3. Okay? At this position, we have already established the AUG, and methionine has been brought to the ribosome. After methionine, the AUG, there's another codon. Remember, when you're looking at the genetic code, these codons are read sequentially. No punctuation, no full stops, no. When you get to a full stop, it means the message has ended. But in between those codons, you read them sequentially. So the sequence is such that the next codon to be read must be positioned in the S site. If this position of this, if the codon is positioned in the S site, it means now we can recruit a charged tRNA carrying what, in this case, if it is histidine, for example, the tRNA that must come into the ribosome must be the one charged with histidine. It can come in the S site. It can't go to the P site because at this point it is blocked. So we are ready to initiate translation. We've now finished making our 70S initiation complex. We can now go into elongation. Elongation means now reading the next codon on the mRNA, codon after codon, going from the 5 prime, going in that direction. So that is the next step. Okay. So what do we need to elongate our our um our polypeptide chain? Now remember, elongation in translation means you already have your uh, AUG interpreted. Of course, the tRNA is sitting in the P side. You want to start now reading the sequential codons. What do you need? Of course, we need the 70S ribosomal unit, chain complex, which we already have. We also need charged tRNAs. And at this point, we know how we got them charged. One other thing that we need are these elongation factors. So initiation, we saw, requires initiation factors, and we know what they're doing there. Elongation has got its own proteins. These are referred to as elongation factors, EF. Elongation factor TU, TS, and G. When you look at them, just like what we saw in the initiation factors, they also play different roles. For example, the tRNA just doesn't come to the ribosome by accident. No, it is brought in by an elongation factor, TU. Okay. In addition to that, the bringing of this tRNA, which is just the ribosome, is an energy consuming process. So once that energy has been consumed, it has to be regenerated. This EFTU has to be regenerated. That means it has to be bound to another high energy molecule. And to regenerate it, it is done by another elongation factor. And also remember, the ribosome is reading the mRNA from the 5 prime to the 3 prime. It has to translocate, move. And that is made possible by elongation factor G. It's also an energy consuming process because to move the ribosome just one codon, you need to input energy in that. So these elongation factors, I'll just refer them to them as EF. They are required for several reasons. So when you look at initiation, it has got its own factors, and you know why they're there. This time around, we want to elongate. Elongation factors also have different roles in elongation. Some of these roles just require the elongation factor getting the tRNA that is charged and bringing it into the A site. Once that is done, that elongation factor leaves the ribosome. Because that process of the elongation factor bringing the tRNA requires energy, when it moves from the ribosome, it has to be activated, recharged with another energy molecule. And another elongation factor, EFTS, comes in to regenerate it. The ribosome, once it has read the codon, it has to move one step. A step means to another codon. Because the ribosome has to translocate or move, it needs an elongation factor to act like a motor. So you can think of EFG like a motor of the ribosome, allowing the ribosome to shift one codon at a time from the 5 prime to the 3 prime direction. Let's look at an overview of elongation. So we can start from here. We have formed our 30S initiation complex. What does a 30S initiation complex have? Let's just look at it. We know that we have got a small ribosomal unit associating with the large ribosomal unit. That is why it's referred to as 70. It's not 50 plus 30. We know why that is. So isn't it? This is just the segmentation fact. Okay. So it's referred to as a 70S. Now, we also have the AUG positioned in the P site. This is where we ended, isn't it? That is what a 70S initiation complex has. This tRNA, this amino acid it is carrying, we know at this point should be methionine, because the codon that was placed in the P site was the start codon for methionine. That is why methionine should be there. Now, because the ribosome in elongation has to start reading these codons on the mRNA codon by codon, it means that in the S site, there is a codon there. And because the S site is no longer blocked by AF1, a tRNA can come in. The tRNA that is going to come into the S site, the amino acid site, must carry an anticodon that corresponds to this codon and the information that is here. In our case, our example was histidine. If this codon is for histidine, it means that only the tRNA charged with histidine can come and stably bind to the S site. Now, keep in mind, other tRNAs will come and float in and try to attempt to bind, but they can only bind if there is a stable codon-anticodon interaction. That's also, so the ribosome is like a checkpoint. It ensures that the stable binding should be complementary. Of course, remember, there might be a flexible point on the third position, but at least it ensures stable anticodon-codon relationship. So now we're moving to elongation. We understand what we have in initiation. Now in elongation, we need to bring in another tRNA to translate this information. I'm going to assume this is histidine. Now, when this tRNA comes into the ribosome, it's going to make a stable, it's the codon on the mRNA is going to associate with the anticodon on that tRNA. And at this position, we're assuming the first codon here is carrying methionine, the next codon here is carrying histidine. I'm using an example from the first slide. It can be anything else. It can be proline or lysine, whatever. But because the example I gave you at first was histidine, I'm going to assume this tRNA is carrying an amino acid histidine. Now, in the P site, remember I said the amino acid in the P site, in this case methionine, is going to form a peptide bond with amino acid sitting in the S site. And it's going to be cleaved, transferring this amino acid from this tRNA to the tRNA sitting in the P site. Now, what we're saying here is that the amino acid, the tRNA, is first of all going to be brought into the S site. This tRNA must carry histidine, if we're using our example, so that the moment it comes into the ribosome, it's going to form complementary base pairing with the codon on the mRNA. Once that has been established, the next step that needs to happen is formation of the peptide bond. This, this the reaction is catalyzed by peptidiotransferase. Peptidiotransferase enables the formation of the peptide bond between the amino acid sitting in the P site and the amino acid sitting in the S site. By sitting in the S site, I mean the amino acid that is held by the tRNA in the P site and the amino acid held by tRNA sitting in the S site. This reaction involves the transfer of this amino acid completely to the amino acid sitting in the S site. And this is what you see here, meaning that the tRNA, the F-met, has lost the methionine group to the amino acid to histidine sitting in the S site. What has happened to this tRNA? It is no longer charged, because remember the charging of a tRNA, it should be binding to what? It should bind to an amino acid. Now at this position, it has donated its amino acid to the amino acid sitting in the S site, so it's no longer charged. Now the ribosome has to translocate. Translocation meaning it has to move to the next codon, because at this point we've interpreted this codon as AUG, that's why we've got methionine there, and also we have interpreted this one as histidine. Now the next message should be we need to bring in these three amino acids to come into the S site. Remember, if this codon is sitting outside the S site, the tRNA cannot come into the ribosome. It can only come into the ribosome because it needs to read the codon. Now, at this point, we have our S site blocked. So the ribosome has to shifting only 3 nucleotides to allow the next codon to sit in the S site. This is what we're referring to as translocation. And like I said, it needs the elongation factor G and energy, of course. So what you see here is the ribosome has shifted, moved this way. So what you see here, again, we have a codon in the S site. The tRNA that has lost this amino acid, in this case methionine, is pushed to the exit site and is taken out, because tRNAs that are not charged do not form, they don't have high affinity of staying in the ribosome. They kind of lose that affinity, so they're pushed out in the exit site. Now, because the ribosome moved 3 nucleotides this direction, it means that this tRNA that was in the S site is going to move to the P site. So all of them have shifted just one codon from the 5' to the 3'. I hope this slide is clear. This is just what is happening generally. You're starting with the 70S initiation complex, where your initiated tRNA sits in the P site. And IF3 is no longer blocking the S site, meaning that we can now read this codon. What tRNA is going to be brought? The tRNA must have a corresponding anticodon to come and bind, meaning it's going to bring in the amino acid that specifies this codon. And that's what you see here. When there's stable binding of this tRNA to its codon, the next step is to form a peptide bond. We know these amino acids are held together by peptide bond. So that the catalysis or the enzyme that is going to catalyze the formation of the peptide bond between the amino acid sitting in the P site and the S site is peptidotransferase, so that we can have a peptide bond. And this is what you're going to see here. The formation of the peptide bond between the amino acid sitting in the P site and that one sitting in the S site involves the complete transfer of these growing polypeptides to the tRNA sitting in the S site, leaving the tRNA sitting in the P site uncharged at this position. Now, keep in mind these two are held together by, there's a peptide bond there. Now, the next thing is that for elongation to continue, we need to continue reading the message that is on this end. The ribosome has to move a codon at a time. So meaning that we must allow the next codon to come into the S site. Why the S site? Because this is the receiving bay for all of these tRNAs to read the information on the codon. It was just different from the initial tRNA, but these other ones that come in all have to come in into the S site. To facilitate this, the ribosome has to shift a codon moving from the 5' to the 3'. So it's not the mRNA that is moving, by the way, it is the ribosome actually translocating, like shifting its position. And that requires energy that is facilitated by the elongation factor G. And this is what you see here. This tRNA has shifted from the A position to the P site. And the one that was sitting in the P site went into the exit site and has been removed. This is what you see there. And this is now the process involving those elongation factors. Like I said, to bring in the charged tRNA into the S site, you need energy. To shift the ribosome, just a codon, you need energy. So whenever you have energy, please use it very well. Don't start spending energy unnecessarily. Okay? This is just a take-home message because we're learning this information just for us to get an A to pass. Very good. But then it's also telling us something about nature. Our bodies don't make their own energy. We always have to input that energy. So when your body has inputted energy, use it well, expend it well. And I mean not just going on Facebook and TikToking every time. Conserve it. If you can't do that, go and sleep or do something else. Anyway, let me go back to my slide. What am I saying here? If this is the scenario on the 70S initiation complex, and I want to explain the same process this time around, I'm bringing in the elongation factors. We needed the 70S initiation complex. It's now complete. Why? Because the 30S ribosomal unit has interacted with the mRNA. The AUG is sitting in the P site. In addition to that, we have our FMET, the initial tRNA complementary base pairing with the AUG. And also we have got our 50S now associating with the small ribosomal unit. In addition to that, our A site is free. If it is free, it can allow for a tRNA carrying its corresponding anticodon to come in. That tRNA can only come into the ribosome if it is charged. And we know the charging process. Amino acid tRNA goes into the cytoplasm, selects the amino acid that this codon is calling for, and then from there, that amino acid is attached to the tRNA on the 3' end. Now that tRNA can come into the ribosome. Now, which site is it going to go to? It won't go into the exit site, no. It will go into the A site. Why the A site? Because it is the one that is carrying the corresponding codon information for whatever amino acid it is carrying. Now, the bringing in of this amino acid to come and sit in the A site requires energy. And this is made possible by a complex of the elongation factor TU forming a complex with GTP and that tRNA. What you're going to see here is that for this codon to be interpreted, for example, CCC is proline. So the next codon that has to be interpreted according to this mRNA is proline. So we need a tRNA to be charged with proline and brought to the ribosome A site. Now, this tRNA, after being charged, doesn't just start walking, running to the A site. It needs to be escorted. It is escorted by who? We are saying it is escorted by elongation factor TU. Elongation factor TU doesn't just come in alone, it comes in with a boost of energy, okay? So it's going to be a complex. Elongation factor, if you look at this position here, you have elongation factor TU GTP coming into the ribosome. So what this means is that to just bring in the tRNA into the S site, you need to spend energy. Okay? Once there's complementary base pairing between the codon on the mRNA in the S site and the anticodon of the tRNA, in this case that brought in proline, this elongation factor can dissociate. Dissociation is facilitated by GTP hydrolysis, like, providing this process with energy too, so that this elongation factor can come out. Now remember, this elongation factor, we still need it because we still need to interpret the next codon. And this elongation factor only escorts this tRNA to the ribosome with energy input. Once you've hydrolyzed it, it becomes GDP. So now, to transform it back to GTP, you need to add in more energy. So elongation factor TS comes in to rejuvenate it. Let me say that again. What we're trying to assume here is that, okay, to bring in hTRNA, you need elongation factor TU. Elongation factor TU does not come into the ribosome also, does not escort this tRNA that you need, or that the ribosome is calling for on its own. It needs an input of energy. So normally you form a complex. This complex is going to have your tRNA, your elongation factor, and GTP. Together they're going to come into the ribosome. Now, the moment the ribosome, the moment the tRNA forms complementary base pairing with the codon, you don't need this elongation factor. Its job is finished. But for it to relieve itself from to let go of the tRNA that it has escorted, you need input of energy, like GTP hydrolysis. When this GTP is hydrolyzed, you're going to have, GDP. Now, this elongation factor, once it is released from the ribosome, it still needs to save another role to bring in the next tRNA. So it needs to be rejuvenated. Back to what? It's binding now against to GTP, because the only way it can escort is if this elongation factor is bound to a high energy molecule. So the sequence repeats. So that's why you're seeing this thing repeating itself. When this elongation factor TU escorts the tRNA and the stable binding, this stable interaction between the codon and anticodon causes destabilization of this tRNA binding to the elongation factor. So it lets it go. In fact, if it's still bound to the tRNA, it's going to be too heavy. So GTP hydrolysis allows for elongation factor to relieve itself, okay, of the binding to tRNA. Now, when this elongation factor goes back into the cytoplasm, it can't work because it doesn't have enough energy, so it needs to be rejuvenated, and it's done, and that is done by the help of elongation factor TS. And this cycle is going to continue repeating itself. So now we can see we've got the AUG in the P site and with the CC in the S site. The next thing that we're supposed to do is to form a peptide bond between the amino acid in the P site and the amino acid in the S site. And this reaction, like I said, is catalyzed by peptidotransferase. This is what I'm just trying to show you here. At this point, this amino acid, this tRNA has been escorted into the S site. So we've got our initial AUG tRNA sitting here with methionine. Now we've got the new tRNA sitting with another amino acid in the S site. A peptide bond is going to be formed between the amino acid sitting in the P site with the amino acid sitting in the A site, of course catalyzed by that enzyme there. This formation of the peptide bond allows for the complete transfer of this amino acid that was sitting in the P site to the amino acid that was sitting in the S site. The moment this happens, this tRNA sitting in the P site becomes unstable. It is no longer charged. And the ribosome only has high affinity for tRNAs that are charged. That is why only charged tRNAs come into the ribosome. Okay, so this amino acid is going to be pushed to the E site by the translocation of the ribosome, one step going in this direction. And this is just what you see here, the translocation of this ribosome. So here, the first thing was the coming in of this tRNA to read this codon. Once that has been done, you have now the formation of the peptide bond, the transfer of this amino acid from the P site to the S site. The next thing is to move one codon from the 5' to the 3'. That is why you see now this amino acid that was in the P site exits because the ribosome has shifted. It is moving this direction. So everyone will move by a codon. So this guy will move here, this guy will move there, and that is what you're going to see, leaving the next codon to be read positioned again in the S site. And because we said the S site is a receiving bay, we can now receive another charged tRNA to come and read this information. Remember, only a charged tRNA will come and sit here, and that tRNA must carry a complementary anticodon to this codon sitting there. Guys, this is about it. I don't know if there are any questions, but the whole process keeps on repeating itself. And of course, we know termination is going to happen when the message termination will happen, guys. Let me use this slide. So the ribosome keep on repeating itself until when the ribosome shifts, okay, a code, and with the shift now position a stop codon in the A site. There'll be no tRNA coming in the A site because remember, the stop codon does not call for any amino acid, that the tRNA won't come in the A site. So the A site is going to be empty. But something is going to come in, and these are release factors. The release factors come into the A site and pretend to be tRNAs. And now, when this tRNA has to make a peptide bond, it's going to completely transfer that polypeptide to kind of like, thinking it's supposed to transfer it to an amino acid sitting in the A site. It won't find anything to transfer. There'll be no peptide bond formed because in the first plate, the release factor didn't bring in an amino acid. So this polypeptide chain is going to be relieved, and then the whole complex is going to dissociate, meaning that the whole message has been read. And if there's another mRNA to be read, again, the complex is going to form and start reading it sequentially. The only thing that I wanted us to mention or be clear about is that in prokaryotes, the process of transcription and translation are coupled, connected. While this mRNA is being synthesized in transcription, the ribosomes are also binding to it. So meaning that on one mRNA, you can have multiple ribosomes translating it. And that is why you can get a lot of gene copies within a very short period of time. Up to this point, guys, I think I am done. I know I've gone a little bit beyond the time I was given. Thank you very much for your patience, but I hope I was able to help for those of us who did not understand translation, initiation, and elongation. Thank you very much, guys. And I think this is going to be our last lecture. Next week, we're going into exams. For those of us who are writing exams, I want to wish you all the best. You've taken these exams before. I'm assuming you're repeating this course. So just take them as exams, just do your best. And for us who are not writing exams, please let's use this time to study. Remember, we've got test ones coming. And also, we've covered quite a bit of information in our previous lectures, including this one. So meaning that test twos and the quizzes will also be coming. So while your friends are writing exams, it's not the time for you to rest. Just take yourself to say, I'm also writing the exam. You're going to have assignments. You might even have labs and all those other stuff. You know how NS is. So I want to encourage you to take this time to, just putting a little bit more energy. We just have a few more months before we close. So we'll stress, but it won't be for long. With that, guys, we'll see you after the exams. Thank you very much for your time. Reach out if you don't understand something that we've covered, including gene expression and other stuff. Otherwise, bye from me. Have a good weekend ahead.
Thank you.
You're welcome. Bye-bye.